Okay, so your class is an intro biology course, and this is during a short semester. So I'm just going to give you right now, before the class begins, a little bit of an idea about what to expect and how to get ready. So one of the things to keep in mind are something to do with dates. So you're going to have access to the class on December 20th. What does that mean? That means that the course information will be open and available to you. You'll be able to see all the lectures. You'll be able to see all the quizzes. You'll be able to do quizzes. You'll be able to read information. You'll be able to so essentially you'll be able to do anything in the class that's part of the class. In addition to that, you should have received a experimental kit, which is all the materials that you'll need to do the lab experiments. Except for the few things that you have to purchase on your own or just have at home. So there's some things like sugar, just some baking yeast that you would get from a grocery store, some hydrogen peroxide, which you may already have. There's a few things that you will probably have to get if you don't already have them. They're very inexpensive and very common items. The rest of the stuff is all within the laboratory kit, and it's stuff that you will find there. You could potentially start doing the laboratories then. The official start date is a week later. So, as far as scheduling the dates for, the lecture blocks are terminated with exams. Okay, so they're going to be 4 exams in the course. Okay, and they're spaced out so that the time that you have from when the course begins to when the course ends, I just took 4 blocks of days, and at the end of those is a day that there's an exam. The exam is going to be available for 24 hours, which means you can do it right when it first becomes available, or you could wait till the very end of that period, and it's up to you. So you have flexibility in that depending on your schedule and what's going on, but it'll have to be done during that time period, and you only have a specific window when you open up the exam. You can't close it, you can't go back into it, you have to complete it, and you'll have a specific clock that counts down, and if you don't finish it within that time, it just cuts you off. So you need to be ready to take the exam. Question, how do you get ready? So there are 4 per exam, and since there's 4 exams, there's 16 total quizzes. The quizzes are there to be sort of graded homework. The idea is, once you get access to the class, you should look at a study guide for a particular block of material. So for exam 1, you're going to have biological chemistry, cell structure, cell membranes. So you should start looking at the lectures on those. There's lectures on all these topics that go through. There's a study guide that has lists of terms. You should start checking things off. Write down the definitions. If there's a drawing, draw a little structure. Just kind of go through them. There's readings that are also links that you can link to the online textbook, and you can open those things up. Use the study guide as sort of just, again, like a checklist. Did I see it? Did I see it? Do I know this? Did I get this? And so forth. Then go to a quiz. The quizzes as homework, they're just open, and you can repeat them multiple times. So you find a question, like, oh, I don't know what that is. Go look it up. That's the point of it. What's the answer? Oh, okay, here, now answer it in the quiz. Hopefully you get them all right, but if you don't, you can repeat the quiz and then do it over again, and it'll give you the average of your attempts, up to three attempts on a quiz. So the idea is you'll repeat these things, you'll kind of look stuff up, you'll remember some of the stuff from the material you've seen, and you'll be studying as you essentially do those quizzes. Hopefully then once the exam day comes, you should be ready because you already did the quizzes. So make sure you keep those things tied together, and then you'll just repeat for each block. After that first exam, you'll have an exam on metabolism of cells and gene expression, so DNA and RNA and proteins. And then we'll move into sort of an organismal block of the class, which still involves some cellular things to begin with. So inheritance patterns, meiosis, mechanisms of evolution, how diversity of life sort of begins. And then we start to radiate out from there to all different types of living things. So we go from single-celled organisms into multi-celled organisms. We'll look at plants, fungi, and animals as we then progress, through sort of the introduction to that stuff and how we get there, how things are classified and all that in exam 3, and then exam 4 is we go through all the different groups of living things, how they're related to each other, how they're unique and different from one another, and you'll continue to go through that with the same, there'll be 4 quizzes per each group and an exam, for each group. Separate from that, you're going to have work that is sort of laboratory based. So you should have received emails with sort of these lists. You can't probably see that from here, but you should recognize the format of this. So there's a breakdown of the different experiments that you have to do. I give you a list of them in order, like in number. And so there's, for example, a seed germination experiment. Should take about 2 to 3 days to do. So you put the seeds in, you follow the instructions. Some of them will germinate, so little plants will come out of the seeds. Once you have those, that experiment is done, but you'll need them to start the next experiment, because then you're going to take those little seedlings and plant them into soil, which you're given in the lab kit in little containers, to start a growth experiment. So then you'll set up a bunch of little seedlings, and then you will decide on certain conditions, which are explained in that lab, whether you're going to give them different amounts of water or different amounts of light, or what is going to be your experimental variable there. That'll go on for 2 weeks. So if you don't start this right from the very beginning, you're not going to have enough time to do it. But if you do start it, you'll have plenty of time. And especially if you start it early, even in the early access week, you'll have more than enough time to do this. But you have to stay on top of it. The other laboratories, some of them will take time in that you have to collect data over a longer period of time, over several days. But it'll take, like, once you get it set up, maybe it takes an hour to set up the experiment, then you'll collect the data. That takes 10, 15 minutes, depending. Some people might take much longer than that, and some people shorter. It really just depends on who you are and how much experience you have with some of these sorts of things. And then you'll come back again the next day, maybe 10, 15 minutes. The next day, 10, 15 minutes, you're going to be collecting data on these experiments. You can have multiples running at the same time. You'll have to keep that in mind. So for these experiments, there are these 8 different experiments. And then there are going to be worksheets, which are separate from them, but they're also related to laboratory type material. So some of these are problems. There are 6 of these. There are things to work through, like genetics problems, identifying stages of mitosis and meiosis, a bunch of population and Mendelian genetics stuff. Classification, so cladistic type 1. So they're not experiments, essentially, where you're collecting data, but they're related to that. There'll be pictures to identify stages of mitosis and instruction on that and so forth, and you have to review images and identify stages within them and count them. So there will be data collection, but it'll be from images as opposed to something alive. The other experiments have living cells. In your kit, you have living algal cells. Make sure you open it up and get them near light, because you're going to be doing an experiment where you're going to count the cells using the little microscope provided in your kit, and you're going to, it'll change over time. That's why we have to let it go for a period of time so you can see that sort of change in it. So you should have plenty of time to do this if you're on it every day, reading through the material, listening to or watching the lectures, performing the quizzes. You spend time each day doing it, and you'll complete it, and you'll do well in it. If you don't, if you let it go to the last minute, you're just not going to have enough time, especially for the experiments. You let these go, and you'll find, like, oh no, that one takes 8 days to do. I only have less than a week left in the class. It's not even possible then to finish. All right, so just start early and work as much as you can. It's largely independent in terms of your pacing. So you can pace how fast you get through it, except the exams are going to be set on specific days at specific times. And you have to take it then during those 24-hour blocks. Right? The other stuff, start working on, get through it as fast as you can, but make sure you go into the level of detail you need, or else you won't do that well, on the exams. But the quizzes should be your main help. So this is just a little intro guide. If you see other information in the course that is not unique to this particular one, it's the same class that's offered during different semesters, just the timing might be slightly different, if it was, say, a summer class or a regular semester course. So just keep in mind, this is specifically for the winter one, and if you see other information, it's probably the same information, except just the timing, I said, will be slightly different.